we mentioned the granulocytes, circulating neutrophils, tissue residents. They are rapidly recruited to sites of inflammation, for example, during an ischemia reperfusion injury and rejection. Engagement of neutrophil pathogen recognition receptors by pathogen or danger-associated molecular patterns after they have exited or left extravasated through the endothelium of the capillary. This stimulates the neutrophils to produce reactive oxygen species, hydrolytic enzymes, and cytokines. The lymphocytes we've mentioned already. So, to summarize, the innate immunity acts immediately on recognition of foreign or dangerous substances through pathogen-associated or danger-associated molecular patterns, or through the lack of expression of cell markers, which in this case are class 1 major histocompatibility complex molecules. Innate immune system mechanisms to eliminate the dangerous substances include the inflammatory response, the natural killer lymphocytes attacking cells with deficient expression of self markers, various families of pathogen or pattern recognition receptors, and activation of the complement system to opsonize, neutralize, or disrupt cell membranes. This is an illustration of how natural killer cells are involved in receptor-mediated cell lysis. The top figure shows a natural killer cell which does not attack a normal cell expressing major histocompatibility complex class 1 molecules. In the example, the picture below, the cells lack these self markers and are attacked by the natural killer cells, causing cell death through lysis. Now we come to the activation of the immune system, the adaptive arm of the or adaptive branch of immunity. The adaptive immune response to an allograft, which is our main concern, but it can react to any antigenic substance, is initiated when graft antigens, including ABO blood group antigens, HLA antigen, the HLA are the major histocompatibility complex molecules, and the third group of antigens that are stimulate or activate the adaptive immune system are non-major histocompatibility complex molecules. HLA is the same as MHC. The major histocompatibility complex molecules are also called HLA, which means human leukocytic antigens, because they were first identified in humans on leukocytes. They are the same as the major histocompatibility complex molecules. In the afferent arm of the adaptive immune response, recipient lymphocytes are activated by donor antigens. When the cell receptors are engaged by the antigens, the cells are activated, they proliferate, and they differentiate into different subsets of cells with different functions, and they send signals for growth and differentiation of other immune cells. In the efferent arm, allograft damage is produced by effector leukocytes that migrate into the graft and by synthesis of donor-specific antibodies. As already mentioned, T cells are of paramount importance. They orchestrate the adaptive immune response. They recruit B cells and other immune cells into the rejection process. Hence, the importance of targeting the T cells by immunosuppressive drugs at the time of transplantation. What about the components of the adaptive immune system? Again, it has cells and soluble mediators. The cells are antigen-specific B and T lymphocytes. By antigen-specific, we mean that each B or T cell has receptors which can recognize only one type of antigen among billions or trillions of potential antigens. Then there are the antigen-presenting cells, short APC, that present antigen to T lymphocytes, because as we mentioned, while B cells can recognize their antigen and bind it directly, T cells require that the antigen be presented to them by a major histocompatibility complex molecule on the surface of an antigen-presenting cell, and we said that the antigen-presenting cells include the dendritic cells, so called because of their star-like shape. These are present in all tissues, macrophage and B cells. The antigen-presenting cells, dendritic cells and macrophage, are a link between innate immunity and adaptive immunity. T cells are further characterized by surface molecular markers. These markers are called clusters of differentiation. Hence, the CD you often find or meet preceding the name of a B or T lymphocyte, CD3, CD4, CD8, CD52, etc. All these are receptors for different ligands. The adaptive immune cell system also has soluble mediators. These are antibodies which are antigen specific, meaning that an antibody will react only with one particular antigen. And these are responsible for the humoral arm of the adaptive immune system. Cytokines are a link between innate and adaptive immunity. They activate both the cells of innate immunity and the cells of adaptive immunity. The T lymphocytes and the B lymphocytes are the main cells of the adaptive immune system. Both of them develop in the bone marrow. The T lymphocyte matures in the thymus, but the B lymphocyte also matures in the marrow. By maturing, we mean that cells with receptors that can recognize and interact with body molecules are eliminated. In the case of the B lymphocytes, the elimination occurs in the bone marrow, hence B cells mature in the bone marrow. In the case of T lymphocytes, the elimination occurs in the thymus. Sorry, just allow me to have a little drink of water. Sorry for that interruption. During their development, B and T cells rearrange their immunoglobulin and T cell receptor genes so that each individual cell has a unique immunoglobulin or T cell receptor respectively. And by rearranging, we mean that the gene for an immunoglobulin receptor or a T cell receptor takes many segments of other genes from other genes, so that it's a kind of combinatorial rearrangement, tabadil wa tawafir. So that each gene has a segment from this gene, a segment for that gene, a segment for the third gene, and eventually the gene will have a different gene which is separate or different in structure, encoding a different receptor in each B and each T cell. This generates enough B and T cells to recognize up to 10 to the power 15 different antigens. Just as for adaptive the immune system, there are signals and receptors that activate the immune system and receptors that activate the immune system. The same exists for the adaptive immune cells, the adaptive immune system. The activating signals in the case of the T cells are specific or antigen cells, which are specific for a particular clone of T cells with a receptor that is adapted to this antigen only. It's like a key in a lock. A key will only open the lock it is designed to open. It will not open any other lock. It's the same thing, key in a lock question. The T cell recognizes antigens from pathogens, or, as we are interested in transplantation, antigens from allograft. And to remind you, an allograft is a graft from one member to another member of the same species, but the members are genetically dissimilar. What about the antigens which stimulate the T cells. We said that the antigens can be derived from pathogens, which I'm not interested in for the moment, or from allografts. There are three groups of antigens that can arise from allografts and lead to rejection. The ABO blood group antigens, the major histocompatibility or human leukocyte antigens, and minor histocompatibility antigens. Before transplantation, we try to match donor and recipient for blood group and major histocompatibility complex. Less often minor histocompatibility complex, but it can happen. The ABO antigens, unlike the rhesus factor, which is expressed only on rhesus positive red blood cells, which anyway are flushed out of allograft before implantation, you perfuse the allograft with cold preservative solution, flushing all the blood out. So the rhesus factor, there are no rhesus red blood cells belonging to the donor in the allograft, and the rhesus factor is not important. The ABO antigen, on the other hand, are present not only on red blood cells, but are expressed on many other types of cells, such as vascular endothelium. So, naturally occurring anti-A and anti-B antibodies, we said there is a blood group A, there is a blood group B, there is a blood group AB, and there is a blood group O. There are naturally occurring antibodies to these antigens. By naturally occurring, we mean that they are present in the extracellular environment without being having previously encountered the corresponding antigen. So, naturally occurring anti-A and anti-B antibodies in the recipient can cause hyperacute rejection of an ABO incompatible allograft. We're going to talk about rejection in a little while. Hyperacute rejection is a type of rejection. A blood group O donor can donate organs to any blood group recipient. This is the universal donor, just as in blood transfusion, because a blood group O donor does not have A or B antigens. So, natural anti-A or anti-B antibodies in the recipient will not react, will not meet the corresponding antigen, they will not engage it, they will not be stimulated to start an immune response. Just as in blood transfusion, blood group AB recipient can receive an allograft from any blood group donor. This is the universal recipient, because an AB group person has no anti-A and no anti-B antibodies, or else his red blood cells will be destroyed. Now, what about the major histocompatibility antigens? These are also known as human leukocyte antigens. These are cell surface glycoprotein molecules that present antigenic peptides derived from pathogens or allografts or virus-infected cells. The T cells, as we mentioned, cannot recognize and engage antigens directly. The antigen has to be presented to them as peptides by major histocompatibility complex molecules on the surface of antigen-presenting cells, that is to say, dendritic cells, macrophage, or say B cells. The HLA glycoproteins are encoded by genes at the major histocompatibility complex locus. Locus is the site on the short arm of chromosome 6. They are co-dominantly inherited. That is to say, you have one molecule from the father, one molecule from the mother, and both are expressed. They are highly polymorphic. That is to say, there are different varieties. For example, the ABO blood group, we have A, B, and O groups. Only 4 types. When we come to classify individuals according to their HLA structure, we have thousands of different individuals, so it is more difficult to find a donor and a recipient who are matching. The HLA molecules are divided into 2 main classes, class 1 and class 2. Both class 1 and class 2 present peptides to T cells, but class 2 present the peptides to CD4 T cells, and we said that CD stands for cluster of differentiation due to molecules on the surface of the cells which allow identification of the type of cell. The T cells are divided into many groups. CD4 positive T cells are the main cells that orchestrate the adaptive immune response, and they recognize antigen or when antigenic peptides are presented to them by class 2 HLA molecules. And class 1 molecules present antigenic peptides to CD8 cytotoxic cells. And to remember the product multiplication, class 1 presents peptide to CD8, 8 by 1 is 8. Class 2 presents peptides to CD4, 2 by 4 is 8. A third class of HLA genes encode other substances such as complement and tissue necrosis factors. HLA molecules are composed each of 2 chains with a groove in between. This is where the peptide antigen is bound in the HLA molecule groove. They have a groove like a cup where the endogenous or the foreign peptide, endogenous peptide is located. The peptide binding groove normally contains peptides derived from self-proteins. Normal cells have self-HLA molecules which express peptides derived from normal cytosolic proteins. If a pathogen invades or an allograft is implanted, foreign peptides replace those derived from self-proteins. The combination of HLA and non-self peptide forms a compound epitope recognized and engaged by antigen-specific cell receptors, starting the elimination or rejection process. And to remind you, an epitope is the site on an antigen molecule that is bound by the corresponding receptor. This picture illustrates the peptide antigen within the groove of a major histocompatibility complex molecule on an antigen-presenting cell. This is the structure on your right, facing you on your right on the surfaces of APC, antigen-presenting cell. They present the peptide antigen to the T cell receptor, which again faces you on the right side when you're looking at the picture. Class 1 molecules include HLA A, B, and C subtypes. They are found on all nucleated cells and platelets, but not on red blood cells. They serve to display endogenous peptide antigens, that is to say those derived from within the cell, such as virally infected or allograft cells, for recognition by CD8 positive cytotoxic lymphocytes. Class 2 molecules also have 3 groups, HLA DP, DQ, and DR. They are found only on antigen-presenting cells. They serve to display exogenous peptide antigens derived from phagocytosed microbes or allograft cell debris that are phagocytosed by the antigen-presenting cells, which also have phagocytic function. And these present these exogenous antigens to CD4 positive T cells. Again, class 2, CD4, 2 by 4, 8. Class 1, CD8, 1 by 8, 8. The subproduct of the multiplication is always 8. The genetic diversity of major histocompatibility complex loci, as we said, there are thousands of variations in the population, is the basis of A, alloimmune sensitization induced by previous encounter to an antigen that may be present in the allograft, and the encounter may be through a blood transfusion, pregnancy, or a previous transplantation. It is also the basis for the attempt to match transplant donors and the recipients according to the number of HLA A, B, and DR mismatches. with a minimum of 0 mismatch, 0, 0, 0, to a maximum of 6 mismatches, 2, 2, 2. Why 2, 2, 2? Because as we said, the HLA molecules are co-dominantly expressed. You have 2 molecules of each type, one derived from the father, one derived from the mother, that are of course different in structure. A greater importance is given to matching at the DR locus because of the capacity of major histocompatibility complex class 2, of which DR is one subtype. These class 2 molecules, when there is a mismatch between donor and the recipient, they will activate CD4 positive T cells of the recipient. And we said the CD4 positive T cells is the maestro, is the orchestrator. They orchestrate or coordinate rejection and recruit B cells and other immune cells into the process of rejection through release of cytokines. Now, the T cells for activation require 3 stimuli. The first is the foreign or non-self peptide presented to the T cell receptor by the antigen-presenting cell. A second co-stimulatory signal requires the engagement of a molecule on the antigen-presenting cell with a corresponding molecule on the T cell. A third signal, as we're going to talk about it soon, is the cytokine interleukin 2, which is released from CD4 positive T cells and other immune cells when they are activated. The T cell, when activated by engagement of its cognate antigen and receiving a second stimulus of co-stimulatory molecules engaging each other, they require also activation by interleukin 2 secreted by the T cells themselves. And this is important because when we are going to talk about immunosuppressive drugs, some of them act by inhibiting the synthesis of interleukin 2 or blocking the receptor for interleukin 2, meaning that interleukin 2 cannot bind to its receptor and therefore it cannot produce its effect. Excuse me, I'm going to have another mouthful of water. As we said, there are 3 groups of antigens that are important in alloimmune rejection. Blood group, ABO blood group, major histocompatibility complex molecules. Then there are also minor histocompatibility antigens. Genes at loci other than the major histocompatibility complex encode proteins with a low degree of polymorphism. That is to say, there isn't such a huge variety of such molecules as in the major HLA molecules. These are called minor histocompatibility antigens. Examples include the male antigen HY derived from proteins encoded by the Y or male chromosome. Rejection episodes in HLA-compatible transplants can occur in the recipients previously sensitized to minor histocompatibility antigen. However, these episodes rarely cause graft loss. They are mild. T cell receptors, as we already mentioned, only recognize antigenic peptide when presented as a compound epitope in combination with an HLA molecule on the surface of an antigen-presenting cell. Dendritic cells are present in all organs, are an important antigen-presenting cell. Other antigen-presenting cells include macrophage and B lymphocytes. Other cells, not normally antigen-presenting, such as vascular endothelium, can be induced during an immune response to express HLA molecules. Antigen-presenting cells have the capacity to 1. Phagocytose and internalize exogenous antigen. possess both exogenous and endogenous antigens, endogenous meaning from within the cell, such as viral proteins. Number 3, they possess the antigen, whether phagocytosed or derived from endogenous proteins in the cytosol, and this processing involves breaking down the antigen into peptides, associating the antigenic peptides with an HLA molecule, and then exporting the compound HLA molecule, foreign peptide, for display on the cell membrane. As we mentioned, the adaptive immune response against an allograft starts when a naive T cell is activated by encountering donor peptides in combination with a major histocompatibility complex, and this meeting occurs in the regional lymph node draining a transplant. What we mean by naive means inexperienced. It has not met its antigen before. This antigen presentation to naive T cells in the regional lymph node occurs in 3 complementary pathways, or occurs through 3 complementary pathways: direct, indirect, and semi-direct. This is not very important. The important thing to remember is that antigen presentation occurs in the regional lymph nodes when the foreign peptide is presented to the cognate T cell receptor. Cognate meaning the T cell, CD4 T cell, with the receptor which is specifically adapted to this particular antigen. We have already mentioned that CD4 cells to be activated require 3 signals. Signal 1 is the interaction of specific CD4 positive T cell receptor with antigen-loaded major histocompatibility complex class 2 molecules on a dendritic cell or other antigen-presenting cells such as a B lymphocyte. Signal 2 is an interaction of co-stimulatory molecules on the antigen-presenting cells such as CD40 and CD80, CD86, also called B7, with these molecules on the antigen-presenting cells interact with corresponding molecule or receptor on T cells, in this case CD40L and CD28 respectively. Signal 3 is provided from 2 sources: cytokines from antigen-presenting cells that direct T cell differentiation into specific subsets, and then interleukin 2 secreted by activated CD4 T cells themselves. This leads to autocrine activation with expansion of T cells and paracrine activation with expansion or activation of CD8 and other immune cells. Autocrine means that the interleukin cells released by CD4, the interleukin 2 released by CD4 T cells, acts on and stimulates the cell of origin, the CD4 T cell. Paracrine means that it acts on other T cells in the neighborhood. The activated T cells then migrate into the graft, causing graft destruction through induction of apoptosis. Apoptosis is programmed cell death, suicide, and also the production of molecules that causes lysis, such as perforin and granzin, which cause the disruption of the cell membrane of antigen donor of the donor cells, causing their destruction. Cytokines secreted by both CD4 and CD8 T cells promote non-specific activation of macrophages, neutrophils, mast cells, and natural killer cells, which also contribute to graft damage, as we have mentioned, and as illustrated partly in this picture.